Good morning, everyone. Welcome to our second edition of the Science of All Proteins seminar for Asia Pacific. Just as we get started, we'll have some more people trickle in. Thanks to those of you who've come right on time. So my name is Dean Powell. I'm GFI Asia Pacific's Science and Technology Specialist focusing on cultivated meat. And for those of you that may be new to GFI, this is your first exposure, we're an international network of nonprofits supporting the development of alternative proteins for a sustainable future food system. And some of those technologies we'll be covering today are cultivated meat and also plant-based meat, actually. So just for a quick bit of housekeeping before we get started, we do have the Q&A feature enabled. So as the presentation is going through, if you have any questions, please add that to the Q&A box, which we'll address at the end of the presentation. And you're also able to vote on questions as well. So if you see a question you'd really like to be addressed, please give that a vote so that we can focus on that at the end. So now for us to get started, I'm very excited to be joined by Professor Hanry Yu. He's a man of many titles, but just to share a few, he's a professor of physiology at the National University of Singapore, group leader at the Institute of Bioengineering and Nanotechnology within Singapore's National Research Agency, A*STAR. He's also the co-lead principal investigator at the Singapore MIT Alliance for Research and Technology. And last but not least, he's also a GFI research grantee. So today he's going to be talking to us about engineering in vitro cellular and tissue models for future food. So before I hand over to Hanry, I'll just do a quick bio on some of his history and achievements. So Professor Hanry Yu is a cell biologist trained at Duke and European Molecular Biology Laboratory, EMBL, turned bottom-up tissue engineer who has created many in vitro models for mechanistic studies and biomedical applications over the last 2 decades. In recent years, he's been translating mechanobiology into useful solutions by developing a framework to study and exploit complex functions. And he has a textbook on that, The Cell as a Machine. Professor Yu has been leading interdisciplinary programs in biomaterials and tissue engineering, biomedical imaging, computation and systems biology, mechanobiology, and recently cell therapy manufacturing. And for today, at Cultivated Meat. He's received A*STAR Scientist Entrepreneur Award twice. He teaches in leading universities in the USA and Asia, advises organizations and agencies worldwide, and has been a handling editor for biomaterials since 2015. We're very excited to have him here today to talk about some of his ongoing work in cultivated meat. So with that, I'll hand over to you, Hanry, and let you take the stage.
Thank you, Dean, for the nice introduction. Let me share the screen a bit. Can you see the screen?
Yep, looks great.
Okay. Thank you. I'd like to thank GFI APAC for hosting this talk. And I'd like to thank the institutions listed here for supporting my work. I need to declare that we have a startup, Hands Innovate, that is actively working in this area. As the host, Dean has kindly introduced me. My lab has been working on creating and using various in vitro cell and tissue model for over 2 decades on complex organs and diseases. One lesson that I've learned is not to model everything, or else we end up creating useless models. In systems engineering's terms, complexity is a measure of uncertainty. If a piece of chicken breast we create has uncertain properties, we will likely throw it away. Therefore, my talk today would start from deep reflections on exactly what we should cultivate and what we should not cultivate in labs or in bioreactors. Is cultivated meat a real piece of animal meat grown in a lab? Yes and no. Yes, because we produce the same animal proteins as in the real meat. No, because it is technically very challenging and expensive to grow a piece of meat from the cells to recapitulate all the structures and functions of the real meat. Therefore, cells are usually grown as raw ingredients. to be structured outside of the labs to become meat by using food technologies. I believe that it will be critical to do something more between the cells and the food. It is critical to precision engineering the raw ingredients, such as cells, into functional ingredients. Functional ingredients are ingredients with defined attributes, such as the cells with strong meaty aroma or 3D printing ink with natural meaty colors, and then assemble the functional ingredient into food. This is like prefabricated homes or car assembly lines. We cannot jump from raw materials or raw parts directly into assembly lines. We need to make functional ingredients or modules like braking or suspension systems. In the cultivated meat paradigm, I show two of them right here. Paradigm number one, stem cells are extracted from animals, expanded into many cells, and then tissue engineering them into muscle stripes or as an ingredient. Then use the food technologies to turn the ingredient into food. There is an exercise muscle here in this paradigm. This is a very old drawing. And this exercise confers more functionality as well as more proteins into the muscle stripes. And the paradigm number two, here, many cells are produced as cell paste and directly structured into food without the tissue engineering of cells into muscle stripe part. In fact, the paradigm number one up there are sort of like a functional ingredient, while cell paste down here are raw ingredients. Muscle stripes are exercised, as I said before, to strengthen the contractile functions of the muscle, and therefore there is a higher protein density when contributing to the texture of the food later. Therefore, it is known that exercised muscle stripe here can give meat a tougher texture than the soft and mushy texture of the cell paste meat from paradigm number two down here. Efforts have been made to construct muscle stripes by tissue engineering, as shown on the left-hand side here, all these muscle stripes. And the papers on the right shows the cells are 3D printed into muscle fibers, and muscle fibers are structured into meat cuts. This bottom-up tissue engineering approach, that is trying to precision engineering the structures of the muscle, tends to be difficult to be scaled up. There is another approach of top-down tissue engineering, which I might explain later, maybe in Q&A. The top-down tissue engineering have some scalabilities, but lack precisions. So it doesn't have extends the cells here. So the problem we face with this kind of tissue engineering approach, which I've worked on for 20-some years, is not just the bioprocessing scalability in the bioreactors and the high cost of tissue engineering. Cell sources are actually a big root problems. For example, we want the cell lines to both divide fast, to generate a lot of cells, and we also want them fully functional. This is not how nature works, okay? Again, for more details, we will explain in the Q&A sessions. An important question here is tissue engineering by artificial muscle or BAM the only way to produce functional ingredients in order to recapitulate the meat functions? The answer is no. Just look at the top plant-based meat out there, where I wouldn't name which one, you know which ones are the better ones. They can achieve about 80% of the meat functions. The rest of the 20% might need to come from animal proteins. Meat functions here are defined as the organoleptic or sensory properties, like appearance, flavors, and texture, not the contractile functions of living muscles, because the meat are already dead. Living muscle functions and architectures are important if you use the material of the muscle cells. So they are sufficient but not necessary conditions for good meat functions. If we do not use the brute force to tissue engineering muscle stripes or bioartificial muscles as functional ingredients, we need to find other methods to impart or enhance the critical meat attributes in the raw ingredients in order to make them functional ingredients. And here, since we're only engineering 1 to 2 critical attributes per ingredient, it is easier to ensure both precision and scalability. So far, the industry is focusing on producing, number 1, lots of cells, number 2, to structure them directly into food. I think it's time to bridge the gap by inserting a step in the middle, that is, to scalably precision engineering of functional ingredient I call SPEFI. This aims to convert functional attributes to raw ingredients with scales and low cost. Let me explain more what functional ingredients or modules are. A functional module is a set of parts, like here on the right-hand side, working together to exhibit measurable emerging properties such as braking. A braking module of a car includes the minimum set of parts for locking the brake disc. A functional ingredient can be 1 or more modules exhibiting the critical functions such as stopping the car. In this example, the functional ingredient can be this braking module here shown in the middle, or paired with electronic control modules. Back to the meat. Meat function is defined by its sensory properties that can be reduced to smaller functional modules such as meat aroma module, which I will elaborate a little more later. To know more about this kind of concept, you can read my book as Dean has explained earlier, Cellular Machine. We dissect the organoleptic properties of meats into smaller functional modules, then creates functional ingredients using the SPEFI approach, and then assemble them to make meaty food. I will illustrate this with 2 examples. One is to generate cellular aroma to be controlled by Maillard reactions and lipid thermodegradation. Number 2 is to generate natural appearance of marbling and meaty color control by decarbonization. It's a mouthful of words. If time permits, I might also share some progress on a GFI funded project. Number 3 is fibrous separations and texture control by dimension reduction. Incorporating this SPEFI approach can enhance the meat eating experience of alternative proteins. The first functional attributes of interest I will talk about is meaty flavors. This is a very complex experience perceived by the brain. To integrate signals from meaty aroma and umami taste. The precursors, reactions, and release profiles, all these attributes and its components together to generate the meaty aroma and umami taste are the functional modules. Taste can be formulated by separate ingredients, such as salt and spices, which are very effective. But meaty aroma is more complex. That's one reason why plant-based food so far still lack that 20% of the attributes. So it should probably come from animal proteins, such as cells. The meaty aroma from cultivated proteins have received mixed feedbacks or are generally weak, mushy, or this type of comments from various sources. It suggests that cell as raw ingredients should be further strengthened or engineered into functional ingredients with strong meaty aroma and improving some texture with animal proteins. We hypothesize here that the insufficient meaty aroma generated in cultivated meat is caused by lower precursor density for minor reactions and maybe some other reactions as well, such as lipid degradations and a few others. Because the ability to seed cells into forming extremely high density and where hierarchically structured meat, like the muscles, is rather difficult. So as a result of that, the number of cells and therefore number of proteins density in the product tend to be low. The second issue is, we hypothesize, is the lower temperature in the center of a structured meat. Therefore, there are incomplete reactions in the middle. The functional modules responsible for meaty aroma is the interaction between molecular reaction and lipid degradation on the aroma precursors such as sugar, amino acids, and lipids. Therefore, it is important to solve this problem by engineering the meaty aroma module involving reactions in the cells. That's another issue which we can address in the Q&A session. In the cells or out of the cells or in the tissue, there's some differences. To achieve the maximal meaty aroma from minimum number of cells, my Siri somehow reactivated. So to achieve maximum meaty aroma from minimum number of cells, then this kind of a natural meaty aroma ingredients can enhance the meaty flavor of various alternative protein products. To test the hypothesis that low precursor density and incomplete minor reaction lipid degradations are responsible for weak meaty aroma from cultivated products, we decided to preliminarily test the hypothesis with a quick fix. If the quick fix works, we probably indirectly test the hypothesis. If we can artificially increase the precursor density, this is a little bit technical. If we increase artificially the precursor density and control the reaction conditions such as temperature and solvent, and if these two interventions can maximize the meaty aroma with the minimal cells, then we can be confident that our hypothesis is on the right track. So we further hypothesize that homogenizing cellular precursors for in situ reactions with efficient heat transfer and precursor mixing can optimize the efficiency of meaty aroma functional module. It's a mouthful. In simple terms, it means suspending cells as small aggregates and homogenizing cellular precursor, partially breaking cells, not totally breaking cells, and conducting reactions directly on the extracted precursors from the cultivated cells can produce cell essence, which is a functional ingredient to enhance the other products, such as plant-based food. Again, this slide is a little bit tricky. It probably involves a lot of Q&A later. Let's start from the cell source. We have developed several lines of myogenic and other cells spontaneously immortalized, adapted to grow in suspension culture, grow to very high densities and reduce the cost for scaling up. I will not elaborate too much here. This is a bulk of work of activities in many parts of the world, and is more or less, to me, is a brute force technical work to get to grow large enough quantity of cells and grow the right cells. We measure the protein contents of the cultivated cells, normalize it to be comparable to the lean pork. We conducted the reactions by suspending cell pellets in plant-based oil, and then introduce heat of the right temperature. Before the reactions, the pellets are white. Under microscopes, they are suspended in oil in the micelle format with less than 150 micrometers in diameter. After the thermal reactions, cell pellets start to shrink in size, probably due to hydration, and turn yellowish brown when forming a byproduct, a pigmented compound that is a typical Maillard reaction product. The figures on the right shows the preliminary data on using enols to indicate there are more volatile compounds generated from cell essence samples, while only a few from the solvent itself. So when evaluated by human subject, they are detecting a strong meaty aroma from cell essence, but no obvious aroma from the solvent. The data shows that at least the cells can generate volatile compounds. To analyze the volatile compounds in a little bit more detail, we search the chemical library and list the compound according to their relative contributions to the overall smells and their individual sensory descriptions. Compounds colored in green here are directly related to meaty, animal-like or brothy aroma derived from the interaction between Maillard reactions and lipid thermodegradation. While compounds color-coded in orange have a roasted, nutty, and toasted smell that are results from Maillard reactions. Meaty aroma compounds, green here indicated, are enriched in cell samples. To further confirm, our cell essence sample has a strong meaty aroma that is comparable to meat samples as we normalize the protein contents between them. We conducted a sensory evaluation. In total, 8 untrained participants were asked to smell cell samples and meat samples in randomized blind order. The questions they were asked include, is there a difference between two samples? Do you think the sample smells meaty? And all participants can perceive a strong meaty aroma in cell samples, and 7 out of 8 couldn't tell the difference between cells and meat samples. The p-value is large enough that we probably can be certain that the meat sample and the cell samples are both meaty and their smells are not different. So this kind of preliminary test is only the beginning. I'm showing you the work in progress, that creating effective functional ingredients for flavor controls is useful. We are further testing the hypothesis and trying to understand the roadmaps to guide our further development of flavor from cells. We notice in the food industry there has been a lot of work of extracting flavors, but dealing with cells is a totally different ballgame. With the strong meaty flavors that cells can generate, not only with the efficient improvement of the availability of the precursors for reactions, but also the control of the degree of interaction between Maillard reactions and lipid thermodegradation, which tends to inhibit each other. When we focus on the specific sensory attributes, of the functional ingredients, we can enable other products such as plant-based products to exhibit strong meaty aroma with as little as 0.5% of the cell by weight, which effectively scale up the amount of final product that the same number of cells can generate by many folds. Okay, I saw there's a lot, probably many questions raised for the short segment I talked about. Now, let's turn to another example for another ingredient. Edible scaffold has been used as raw ingredient for plant-based or top-down tissue engineering of cultivated meat for a long time. In fact, it's still very popular. To turn this raw ingredient into functional ingredients to look like real animal meat, we reduced, again, the organoleptic properties into appearance, flavor, and texture. The appearance can be engineered from marbling and coloring module itself. We want to know what plant fruit have the texture appearance looking like marbling to start out with. So we train the neural network with lots of marbling meat images. We use transfer learning of AI to optimize the algorithm and reduce error. We look for texture similarities to marble meat, as well as the phenolic content, and discover that jackfruit as well as brinjal have both properties. Here is, when we look at the color, it's known that the color of jackfruit simulates that of the meaty color. So we look at the color carefully, and it seems that most are found on the cell walls of the plant. We hypothesize that the intracellular polyphenol can be released to bind to the cell walls and oxidize to confer the optimal colors. Usually, again, there's some details here. It is interesting for us to discover if you don't incubate them after you break the cell wall and cell membranes, the color will not be so optimal. So the marbling is the result here for the inability of the polyphenol to bind to lignin. The mechanistic insight is postulated here. And so, again, taking an explorative approach, we have developed interesting ways to decompartmentize the cell contents for them to interact with each other in circumstances they normally don't interact. Here is to try to show that by combining the color control through decompartmentization with decellularization using sodium dodecyl sulfate, which is a detergent, to solubilize the compartment, we break the wall, break the membrane, and mix them together artificially over a longer period of time, using various physical methods, incubating with different extract with the cellulose on the cell wall, which confer different color control for different meat. So the picture in panel D here shows that the comparison between different kinds of meat with different kinds of plant-based materials. We add cells to this kind of scaffold, grow it to conference, and see if the cells will mask the scaffolding coloring. It doesn't. So the coloring is preserved where the cell has been growing in the scaffold. We cook them in water and in oil, and the colors are permanent. It can be modulated with pH, but depending on the conditions of heating, they are more or less permanent, and it can be controlled to different degree. This is simply the videos of showing the color changes, where tend to be minimal. It's quite stable in water and in oil. This is another video of a bacon-like plant-based meat analog. It tends to brown like the real meat. Yeah, you will roughly get the sense of what it looks like. Okay, let's come back to the discussion here. I have so far used two examples to illustrate how scalable engineering, precision engineering of functional ingredients can be done. Unlike the paradigm number one, we do not use brute force to grow up contractile muscle fiber bundles, which is a very important effort. I still think the community will continue to do those hardcore tissue engineering work. But it's very challenging. It will take quite some time. We spend much effort to precision engineering the cell paste and other ingredients into functional ingredients with optimal cell or non-cell based sensory properties in scalable ways. Regardless whether you're making plant-based food with 0% cells or cultivated meat with 100% cells or hybrid with some cells, the key proposition here is we need to do something in the middle, okay? And you might think that I'm talking about hybrid work here. In fact, I don't think the hybrid is a meaningful term in this discussion. Practically and scientifically, all cultivated meats are hybrid. Even the most scaffold-free cell sheets cultivated methods will not produce 100% cultivated animal proteins. As I explained earlier, I didn't show the data here, that if you have taken a cell pellet, which is 100% more or less, then you just make a chunk of it and directly cook them. The texture and the colors, there's no color actually. And also the flavor sensory-based sensory functions are almost totally off. Okay, so we need to do something while the industry are solving the cell manufacturing scale and cost bottleneck problems. Our dream is for the alternative protein products to appeal to the bulk of the population hopefully even the meat lovers. These are some of the examples of the product that benefit from such an approach. And okay, so I guess I still have about 5 minutes. I can talk about, so Dean, should I talk about the GFI project?
Yeah, definitely. Please go ahead.
Okay, 5 minutes, right? Okay, let me see. Yeah, first, thank the people involved in the work in different phases, and the collaborators and the funding support. So now this is the part which is a bit also very preliminary. The texture perception is another important function to replicate. The interaction among food structure, oral processing, and mechanical properties all decides the texture perceptions. So one issue, as we survey through breakdowns of this kind of processes into different parts, we identify there's interesting property on fiber separations as a critical attributes to be worked on. So separation of fiber bundles upon chewing influence the perception. The force requires the cells requires the fibers to which influence the mechanical properties at different stages of chewing and such. So it's with the ingredients such as the soy essence, which I talked about earlier, we can probably engineer textures with other materials as a base. One good example is HMMA. That is high moisture extrusion type of the materials, meat analogs, which are pretty good mechanically with mechanical properties and have some microstructure, microscale fibrous structures. Although HMMA has recapitulated part of the meat mechanical properties, its fiber separation behavior is not like the real meat. Extrusion gives HMMA molecule orientations and layer structures so that HMMA can show fibrous appearance under tearing vertical conditions to its molecular orientations. However, if HMMA is sliced into sheets or torn, it cannot show fibrous appearance like meat under the same treatment. You see on the left-hand side here. This is because the HMMA in the inner side of each layer, fiber bundle concept do not exist. Each protein molecule is too weak compared with the force linking them. So when the force is applied to the layers, HMMA will be broken before its fiber separation. So is it possible for us to engineer the fiber separations for HMMA to improve its fiber separation properties? And the answer is yes. Let me see. Is this a video here? Yeah. So just show you. I forgot to turn on the video. So you can see the you can see when you cut, the fiber separate. And typically HMMA, when you cut, it's just cut. Depends on how you bend it. Okay. Right. So here as shown, what we propose to do is as illustrated on the top. The HMMA sheets are first derived from extrusion, extruded bar. We slice them, and by imprinting, by reducing the third dimension as a whole block, because it's rather difficult to have a whole block of materials to be structured with fine precision. So this is where the building simple models on complex target comes into play. One typical method is to reduce the dimension of the 3D structures into 2D slice. And by using the imprinting methods on the slice, we can actually, there's some tricks here. For example, we have to perforate this in order to make the mechanical bond weaker between the materials of adjacent strand. And then we try to stack them up together, and then to form the meaty and the fibrous like meat cut structures. So you see the cut fibrous separation. When you cut, the fibers will separate. And the tearing, or as well, you notice. The fiber will separate in different, so the so-called long range and isotropy is observed, like in the real meat situations. One question people always ask, later we can also discuss, is when you reduce the 3D into 2D, you seem to have added additional steps into the middle. Is that scalable? So scalability seems to suffer a back track. But actually, when you reduce the 3D into 2D, the scalability increases drastically. And then we put together we work quite a bit on the binder and fat, and combined with fat replacer, and to construct into meat cut like prototypes. And one thing is that this additional steps of putting binders to put the 3D structures together gives us rooms to manipulate what's going on in there, such as the color, such as the texture, and adding fat or not adding fat. So it enables the door to be wide open. And it solved a couple of problems with HMMA. One of the problems with HMMA is you see this cooling die. Eventually, it restricts the thickness you can have. Of course, by stacking them, the 2D sheets into 3D meat cut like construct, you can stack as high as you want. And the other issue is obviously the percentage of the fat you can incorporate into this kind of construct. There's a limit how much the fat the HMMA can tolerate. It has been reported that more than a certain percentage of the fat, the structure of the fibrous structure will disappear. Okay, so for example, it's shown here. The structure will disappear in higher concentration of the oil content. So that's it's you can do many things. And the good part, especially about this, compared with the existing approach, is when you work on a 2D sheet, it becomes quite scalable. Using roll-to-roll kind of imprinting methods, you can manufacture it in a semi-continuous fashion and with high degree of precision. This is an instrument which can have nano imprinting even. Okay, so micro imprinting is enough, or even sub-millimeter printing is enough, and isotope can be created with different degree of imprinting technologies as well. So this kind of intermediate functional module effort and functional ingredient effort would probably open the door for many possibilities. Yeah, I think I will stop here. Thank you for your attention.
Great, thank you so much, Henry. It was a really interesting presentation, and I really appreciate the breadth of work that you're doing and were able to share with us. I think having a focus on the functionality of the ingredients is really important when a lot of conversations in this space are really focusing on getting those costs down and just trying to replicate the cultivation of cells and formation of those muscle fibers, when in the end, you're right that we do have to get the cost down, but the taste has to be right as well as the texture. And people don't just want to eat cultivated meat because it's meat; they want to eat it because it tastes great. So, as I mentioned at the start, for those that might have joined a bit late, we have the Q&A box function enabled. So, if you have questions, could you please just add them into there so that we can address them and others could vote on them as well. I saw a few people had their hands raised. If they were wanting to ask questions, we will just have to use the Q&A box feature. So please just add them in there. And same for those that might have put things in the chat. So we have one question from the chat, which I can read out to get us started. This is from Nicholas. So they say, Dr. Yu, you described extraction of membrane phospholipids from cell membranes using a plant-based oil solvent. Presumably, this can be from any cell type, since all cells have membrane phospholipids. This approach appears different from incorporation of whole adipocytes or adipogenic cells into the product described by others. What are the relative advantages for lipid incorporation as described by your method?
Yeah, thank you. The answer to this question is it works, and it's complicated to exactly pinpoint what's the reason it works. And I think, based on the hypothesis which I had, the two-prong approach caused the initial total incorporation of adipocytes or other things or muscle cells into the food is because the accessibility of the maillard reactions or lipoxidation to and the control with them to in the bulk setting. So when you solubilize or suspend them into oil in the presence of some water, okay, because they're in the micelle format, so therefore they more or less distribute the cell to be accessible to almost all the reaction when you heat it up. If the pellet size is not big enough, the uniformity of it will have better control of the reaction. Now, there's a lot of things which we don't understand. For example, if you pre-homogenize the whole thing, that means you break apart the wall of the cells. Actually, the flavor actually got greatly inhibited. So we hypothesize that's probably because the interplay between the lipid and the Maillard reaction, somehow compartmentalization of those components in the cells plays a significant role in doing this. So there's a lot of things we are still in the middle of testing. I have a PhD student who works on the flavor of cells. And so I'm hoping that to give you more answers to your question next time.
Thanks, Henry. We have another question here looking at color. So is there a way to increase myoglobin synthesis in the cultured cells to give a more natural color to the meat products?
Before, let me say a little bit more about Nicholas' questions about what's the advantage of it. Well, the major advantage of the cell essence technology is really because the stronger flavor of the of the of the meaty flavors, aromas can be generated from the cell essence. So I mentioned briefly, effectively we can only incorporate 0.5% to 2% of the cell mass to equivalent to a plant-based product, and it will give a very distinguished, very strong meaty flavor. So which effectively makes the cost drastically down for the hybrid product. So even though I hate the term hybrid, which has a very weird connotation, but the reality is that if you use that kind of approach, you can supplement even the higher percentage of the cultivated meat product. So that's the major advantage, I think. As for the mechanisms, we're still struggling to understand.
Okay, understood.
Yeah.
And I think that's very exciting. I feel like I haven't heard many other people talking about such low inclusion rates of muscle cells to influence flavor by so much. So that would be very exciting once you're able to publish more information around this.
And also there are the different, when you concentrate the meaty essence like that, and the differences in terms of muscle cell, fibroblast cells, it tend to be less. I wouldn't say it's less important, but it tend to be masked somewhat, because if you have a strong flavor, which can more or less, people treat it as a meat flavor. That's probably good enough.
Very interesting. Yeah. We have another question here from Marie Cruz. You mentioned that no one can achieve 100% purity of a 100% pure cultivated product. There'll always be some other elements to it. So they were wondering what the other byproducts are. If you could answer that one, thanks.
Right. So there are various ways to make so-called 100% tissue product. And most of the times, the cells can be only growing in scaffold because, as I explained earlier, the cells, if you want them to grow in larger quantities, the best way is to grow them in suspension cells. And when you don't, when they can suspend, they usually would lose the ability to differentiate well. Now, so in terms of what other materials, so depending on the configuration you're growing, most people will have some kind of microcarrier or scaffold materials in the product itself. Even if you remove them, you still need using structural protein or binders or fillers for the food product itself. Because let me give you one example. Whatever you're culturing usually don't have color. Usually it's opaque or translucent or even transparent in some cases. Then you have to add color to it. So it's very difficult to have 100% cell mass without anything else. Yeah, of course. In fact, there's a very interesting debate going on. That is, whether you should use cells to control those organoleptic properties, or you should use non-cell components. In fact, for people who try to gain the texture by having a lot of cells, they reduce their degree of freedoms to control it with other means, such as plant-based or fermentation-based product. So you give and take, depends on what you want. So what I say is that for ingredients, we need to have a variety of ingredients of different sorts in order to gain the ability to control functions. So that's where I see the industry should be going forward. Because if you tap into the entire field of alternative protein industry, instead of just narrowing down to have pigeonhole in the cultivated meat industry, which is many people believe is a niche market. That's because if you focus on product number one, then of course it's so hard, it's so costly. And having worked on tissue engineering for 20 some years, I haven't seen any successful larger bottom-up tissue engineering approach really reaching the clinical market. There are a few, but not at low cost enough and scales. For top-down tissue engineering approach, it's really the density, the structure is lacking. You just dump cells into scaffold. Usually you don't have control over the details. So what I'm trying to say is that all this work needs to be going on concurrently at the same time if we have to reach the final target we want to have.
Great, thanks. And I think that's an important point about not getting too focused on the cultivated or plant-based label. And I know you don't like the term hybrid, but yeah, I think that's really going to be the most flexible way forward to get the best products out there. Our next question, kind of, you were just answering a bit then, but it would be interesting if you could expand a bit more. This is from Jennifer Young asking, they want to get some more of your thoughts on the top-down versus bottom-up approaches and what you think is the best way forward. Do you think that top-down might provide some specific advantages for certain meat properties around scalability, costs, etc.? So maybe if you just build on that last point you were talking about.
Right. So most of the tissue engineers, including some very big names in the space, they're all starting from the top-down tissue engineering. Top-down tissue engineering basically take a scaffold which looks like the final things, such as a bone or cartilage, or in this case, a piece of meat look like. You structure the scaffold in such a way, and with a porosity inside, and then you try to see the cells onto it. But the problem with top-down tissue engineering approach, as criticized by bottom-up tissue engineering approach, is always that it's like you make a building with scaffolding, with the pillars there without rooms. So you don't have a comfortable environment for the cell to sit in, or structuring exactly what's the hierarchical structure of the muscle, which I showed you earlier in my slide. So you are at the mercy of whatever the cell interact with each other naturally to do it. Is it possible in the long run to merge these two? There are already efforts who have, to some degree, have been successful, which you learn from the work of decellularized space. You take an organ, use detergent to decellularize it, so all the intricate structure seems to be remaining for the ECM, and yet you still have the larger structures. To some extent, it works partially, not totally. Okay, and there are people who have been using middle approach between top down and bottom up. That is to build a centimeter long piece of stripes by using, I know there are certain people using arginate or whatever other materials to form a tubes, or there are people who use the partially top up, partially bottom up, partially top down, by using bioreactor, using hollow fibers and such. These are roughly the top down approach. But retrieving them is a pain, and also you probably, again, you still have to try to make them into a larger food. So it's still work going on, I guess. So personally, I don't. Okay. The issue becomes more complex if you don't care about the intricate structures of using the bottom-up approach. Then isn't it true having a top-down approach will give you better ingredients. Yes, it might be true, because using top-down approach, if top-down approach give you roughly that kind of texture you need, using a centimeter strand, or then it might work. But to keep a larger construct itself is extra effort. So whether people want to go through the extra effort, is it easier to use different methods? That I cannot say. So maybe it's possible somewhere in between a top-down approach and the functional SPFI approach, SPFI approach, combining together would make it work. So I'm still open to that.
Oh, great, thanks. I think it's an interesting area to continue that conversation on and kind of break out of these different molds that people put themselves into. Unfortunately, we've run out of time. We had some other good questions there I would have loved to have you answer, but we're going to have to wrap up. For those that might have more questions, you can find Hanry online on LinkedIn or other places and maybe reach out to him. You can also contact us at GFI if you have any questions for us as well. And we will also have this recording available on our YouTube channel afterwards. So for those that were asking about accessing the slides or presentation, give us a couple of days and we'll have that uploaded there that you can share with other colleagues that might be interested. So thank you all for your attendance, and thank you very much, Hanry, for a very interesting presentation. We really appreciate your time and sharing your expertise with us all here today. So thank you.
Thank you.
Excellent. All right, everyone, have a great day. See you all later.
Bye-bye.